before Dr. Stein gives his presentation. All right, so what I want to do is actually tell you a little bit as a follow-up from what you've already heard in terms of the genetics behind some of these problems that you're hearing about. So I'll tell you a little bit about our current working model, and in the end I'll tell you a little bit about some of the future directions, namely in particular how the skin biopsies that many of your children have had, I think, have the potential to lead us down some very exciting roads. All right, so a lot of you have heard me give this talk before in some form, and for the new families, I hope this will be well received. So kind of historically, as you know, my original interest was in the heart issues, but it became clear that we could probably use some of those same kinds of approaches, meaning define whatever the clinical problem is, the clinical phenotype, and then go back and try to define the genotype or the genetic cause, and see if we can use that same approach like what I talked to you about yesterday for finding the heart gene to try to identify some of the genes that we think might be contributing to the cognitive and behavioral problems. And so these are kind of the questions that I came up with early on and continue to address. And first, that is, are there genes in 11q that are involved in brain development and function, which is obviously what you would predict? Is there a correlation between the presence or absence of any of these genes and the degree of cognitive ability or the behavioral problems? Can we, just like what I showed you for the heart story, use animal models to try to understand the role of these genes in human disease? And later on, I'm going to show you some unpublished data that has been graciously provided to me from one of my collaborators that will give you some insights into that. I do ask that we're actually not going to show that on the live stream because out of respect to my collaborator, but I will provide those here in the room with the opportunity to view that. And then finally, are there any potential treatments or therapies that could be developed based on what we know about the function of these genes? So the aims are, one, to perform high-resolution mapping, i.e. using the array CGH that you've heard so much about now, to get a readout of the specific genes that are deleted, and to correlate that with the clinical phenotype, which you've heard already a lot about this morning, thanks to our colleagues here, Sarah and Natasha and others. And then based on that, to begin to identify potential candidate genes and what their function might be and how they may contribute to that phenotype. And then lastly, to look at animal models for studying the mechanism of how those genes function normally and what might happen as a result of the loss of that function. And then lastly, to go to your children, and I will never ask to do a brain biopsy from your child, but we can do something almost with the same scientific power, and that's what the skin biopsy thing is all about, and I'll come back to that. So today what I'm going to talk about first is how we've approached this to identify some genes that we think are contributing to the varying degree of cognitive impairments. And then I'm going to specifically tell you about the approach we've taken to identify what we think is at least one gene in 11q that's probably contributing to some of the autistic features that Sarah and Natasha have identified. So, this is probably not newsworthy to you, but the fact is, over the years, and this actually started back with Zona Lai that some of you may remember all the way back to 1998, I think, in 2000, and you didn't even need to do any formal testing to at least qualitatively appreciate if you just go next door to the room, and you can, I think, pretty easily appreciate that there really is a range of cognitive function among your children. And so, as you've already heard, we had some sense that there was probably, and intuitively that's not too shocking, a correlation between the deletion size and the degree of cognitive impairment. And on the next slide, what I'm going to show you is, I'm going to just cut right to the chase and show you what our current thinking or model is, at least, for the number of genes in distal 11q that we think are contributing. And there are probably more, so this is an underestimate for sure. One of the disclaimers that I want to make that I continue to be humbled by all the time, Sarah and Natasha might disagree with me a little bit on this, but I'm among friends here, and that is sometimes we tend to be very analytical in how we come up with these assessments to quantify your child's level of functioning. So if your child comes out with a full scale IQ of 45 or 55 or 65 or 75, we kind of lock onto that in certain ways, and certainly there's a lot of value, obviously, about that. But I think I mean, I'll just share with you my own personal feelings about this. You know, when we heard Tristan sing the other night, right after that, I went up to Tristan, and this is the honest to God truth. I said, Tristan, I could never do what you just did. I would never have the cognitive ability to remember the lyrics of a song and stand up and sing for 5 minutes. And, you know, Tristan has a pretty good-sized deletion, so I think the message I want to send to you is, yes, there's great value in trying to quantify all these things, but let's not lose perspective of the whole picture. So now I'm going to move ahead to show you what our current model is, and I know that this is a very busy slide, but let me kind of tell you about the extremes, and then I'll show you how we've gone about kind of refining this. So a couple of things to start. Intuitively, what we've clearly actually found is that for the patients that we've assessed that have the highest IQs, which are basically in the low normal range, high 70s, even I think low 80s in some cases, without exception, those individuals have the smallest deletion. And that's not too surprising, okay? And then kind of on the opposite end of that is there's nobody with the largest deletion that has the higher end of the scale of the IQ. Does that make sense? So nobody with a deletion size of over 12 megabases has tested to have an IQ, let's say, of 70. And likewise, anybody who does have an IQ of 70 has a deletion size that's probably less than about 10 megabases or so. So that's pretty intuitive, and our studies have, without exception, demonstrated that. The other thing that is now clear is, again, as the deletion size gets bigger, the degree of cognitive impairment gets more severe. But where we've really most recently made some progress is by studying some of the children that have these interstitial deletions. And basically, what we found now is that there appear to be at least, and there are probably more, 4 regions in chromosome 11 that are deleted in some or all of your children that contribute to this overall phenotype. And what it comes down to, and so if you look here, so if you just have a small deletion to the end of the chromosome, you're most likely going to be relatively mildly affected. If you have a small interstitial deletion either here, or either here, just by itself, spanning that one region, you're also relatively mildly affected. Where it gets a little more complicated is when you have multiple regions deleted. So again, the extreme is if you have the big deletion that covers all 4 of these regions, you're going to be most significantly affected. What we found is that this region 1, again, by itself, appears to be such that if that's just deleted by itself, you're going to be relatively mildly affected. But if you have a deletion that spans region 1 and region 2, so probably 2 genes, then you're much more significantly affected. Or if you have an interstitial deletion that spans regions 1, regions 2, and regions 3, then again you're going to be more significantly affected. And again, taking the extreme, if you have all 4 regions deleted, then you're going to be most significantly affected. If you have region 3 and 4 deleted together, so what I would refer to as a small to medium-sized deletion, or if you have just 4, 3, and 2 deleted, but without 1, you're going to be somewhere in kind of an intermediate range. So it's fairly intuitive. The bigger it is, the more significantly impaired you are, the smaller you are, the higher functioning you are. If you have just one individual locus alone deleted, you're relatively mildly affected. And it's really the combination that helps you to predict, we think in most cases, the degree of cognitive impairment. And really what I'm just referring to here is full-scale IQ. And this is just basically another way of showing that. So I've sort of qualitatively defined the deletions as being small, medium, and large. And what you can see is that there is a huge drop-off once you get past this 12 megabase breakpoint, where the full-scale IQ significantly drops to somewhere in the range of 40s. So this is kind of to summarize that concept. All patients that we've tested that are over 5 years of age are mildly impaired, that can be mildly impaired. Wait, let me say that again. All patients over 5 years old that are only mildly impaired have a deletion size of 12 megabases or less. And another way of saying that is no patient with an IQ over 60 has a deletion of greater than 12 megabases. And that's roughly based on a fair number of patients now. So what that tells us is somewhere at that 12 megabase region, there must be a critical gene. That's kind of the cutoff. And so now we can use the power of the Human Genome Sequencing Project, again, to see what's in that region. And it turns out there is a gene there called this BSX gene, which is an abbreviation for brain-specific homeobox protein, which again is always reassuring when you're looking for a gene that you think has a function in brain that has the word brain in its name. And it turns out there's an investigator in Germany by the name of Matthias Trier, who unfortunately couldn't speak at this year's conference, who spoke 2 years ago, who told us some of the work that he's been doing. And I can tell you that when he makes a knockout of this gene in these mice, when you knock out both copies, now remember your child has one copy at least, or two, but when you knock out both copies at least of this gene in the mice, they are severely cognitively impaired. So moving right along, when we look at some other parameters of cognitive function, so digit span, which is really just kind of a short-term memory, I tell you 5 numbers and then 5 minutes later I ask you to recite back those 5 numbers, which I would probably fail. But in that particular aspect of cognitive function, we see a big drop-off between those patients that have a small deletion versus those with a medium, and then again those with a large deletion. And kind of using a similar approach, we identified a gene that seems to be critical, contributing to that selective cognitive impairment. And this gene that you've heard me talk somewhat about in the past is called neurogranin. Basically in mice, the more neurogranin, or the more of this protein there is in the brain, the stronger there is for signaling, for learning. And we actually think that neurogranin is likely a molecular mediator of environmental enrichment. And that's actually extremely interesting because it turns out there are other situations, pathological conditions, where for whatever reason, when there's memory and learning impairment, the level of the neurogranin protein in the brain is diminished, specifically Alzheimer's disease. So we think actually what we learn from your children about neurogranin could have much, much broader implications for a much larger group of patients. So neurogranin improves the ability to create memory and long-term memory. And if you delete the neurogranin gene in mice, they have decreased so-called long-term potentiation. They have a decreased ability to retain and recall memory. They have decreased cognitive ability, and they're actually very anxious. And in fact, one of the people who's been doing the most research on this at the NIH told me it's actually very difficult to study these mice, and they have this whole battery of cognitive testing because the mice are kind of all over the place. They're very anxious and they're hyperactive. So that suggests this gene could be contributing to some of the ADHD phenotypes that you have been found to have. And the other thing that I think is very interesting is, just like human beings, mice do well when they're in a much more enriched environment, kind of shown in this slide. And so we give them toys and lots of space and exercise wheels, for example. So if you have a mouse go for a 2 or 3 mile run every day, their cognitive ability improves, and it's been shown that that coincides with an increase in the level of the neurogranin protein. Now, we haven't proven this yet, but what we think is that from these studies in the mice at least, the more neurogranin you have, or the less neurogranin you have, that correlates with your memory and learning ability. So we think that neurogranin actually may be a very critical player in mediating this long-term potentiation and learning. So one of the things that in the future we would love to be able to do is come up with ways to boost up neurogranin levels, not only in your children, but what about in someone like my dad, who has Alzheimer's disease at age 89, who's basically losing brain cells every day. And so, you know, this isn't going to be a cure for Alzheimer's disease, but what if you could take those cells that are still intact and ask them to work a little more? So boost up the function of those that are still preserved. So that's, I think, a very exciting potential therapeutic opportunity that could come from this. And in fact, it's, I think I already told you, been shown that in the brains of some patients with Alzheimer's disease, the neurogranin levels are actually diminished. But what we still don't know is increasing neurogranin by itself necessary and sufficient to improve memory and learning? And we actually are embarking on an approach to determine that, and that is we can overexpress neurogranin specifically in the brain of a mouse. And there are mice that are genetically engineered to have some of these memory and learning disorders like Alzheimer's disease. So we can now take a mouse that has a well-defined genetic model for memory and learning problems, and then we can introduce in this neurogranin protein, as shown in this gene, or in this slide. All that orange is the neurogranin protein, and we can use this kind of cartoon idea to artificially, through genetic trickery, turn on the neurogranin protein in the brain and see if we can then rescue that phenotype. And this is, I'm just going to skip this slide other than just to show you that we can actually use that vector to show that we can express, at least in cells that are cultured, neurogranin sort of artificially. So that's kind of the basis for what we would like to do. Okay, so in the second half, what I want to focus on then is what you've already heard about to a large extent. I don't think I need to tell you to show you this slide again. But from the genetic standpoint, so about half of the kids, as shown here, that were tested very rigorously, fulfill the diagnostic criteria for autism spectrum. And interestingly, there was no correlation between the size of the deletion and whether or not the child tested for the spectrum. And so even the child that was tested with the smallest terminal deletion, namely 8.7 megabases, was found to be autistic. So just like what you've seen in some of the previous slides, what we could do then is, so if you look up here, that's the child with the smallest terminal deletion of about 8.7 megabases. And there are actually, is that the 8.7? No, well, okay, so there was a child with an 8.7 megabase deletion, and then we subsequently learned about a child with an even smaller terminal deletion who actually didn't really fulfill all the diagnostic criteria that we have for Jacobsen syndrome. Oops, did I just lose this? Is this still working? Or, there we go. But actually was found to be autistic. He was diagnosed with autism first and then had the genetic test that revealed this relatively smaller terminal deletion. So that suggested that maybe at least one gene for causing autism could be in this region. And then we knew of a couple other children that had Jacobsen syndrome clinically, including the diagnosis of autism that had an interstitial deletion, which suggested that if it is one gene, that maybe it's within this region of overlap. And then most recently, we came across a rather extraordinary patient from the UK who was diagnosed with some kind of degree of intellectual disability. Because of that, she had an array done and was found to have a very tiny interstitial deletion containing just a few genes that happened to be within this region of overlap that we had already defined, which was actually within the region of overlap of another patient that had been previously reported to have autism. And so we didn't know anything at all about whether or not this child might fulfill the criteria for autism until Natasha, I think, conducted a 3-hour interview with the family over the phone, very rigorously. And in fact, that child as well fulfilled the diagnostic criteria for autism, or autism spectrum. And when you look closely at that very small region, there are only just a few genes. And one of them happens to be a gene that is already well studied, in particular by a group from Japan that I am now collaborating with, a gene that has a few different names. But basically what this gene does is it actually regulates the formation of synapses or neurons, how they connect through some very complex biochemical pathways. I'm going to ask Tim now if he could cover up the video, because this is quite privileged information, but I am happy to share it amongst you guys here in the room. And so I originally contacted the group in Japan that's been studying this gene about 2 years ago, because they had already shown that if you specifically delete this gene in mice, they do have some selective cognitive impairment as well, which kind of goes along with the fact that many children with autism have cognitive impairment. But they had never thought about the possibility that this gene may have a role in autism until I approached them. And so, coincidentally, I happened to be invited to come to a cardiology research conference last summer in Tokyo, and I got the opportunity to meet with these investigators. And what they were graciously able to do then is embark on a very systematic study of their mice to determine whether or not, at least in the mouse world, they might exhibit any features that were reminiscent of autism in humans. So let's see if I can start this. And so on the left is going to be the normal mouse, and on the right is their knockout mouse. I don't know why. Oh, here we go. All right, so there's the normal mouse. He's pretty interested in not just those marbles, but also his surroundings. So periodically he looks up and around. And then if you look at the mouse that's missing this gene, I think you'll begin to appreciate a difference in the mouse's behavior. Now, I've been told that some of us researchers look much more like the mouse on the right because we're very repetitive and never have a clue as to what our outside world is like, right? So I think you can begin to get a sense that this is just one, and certainly mice are not humans. Oh, here, now you can see it side by side. Maybe it's a little more useful to look at it that way. So the one on the right is really kind of obsessing on burying these marbles, and the one on the left is acting much more like a normal mouse, looking around, trying to get out. And if this works right, they can actually quantify these behaviors, believe it or not. So they're videotaping, and what they clearly show is that when you look at the wild type compared to the knockout mouse has buried a lot more of these marbles. And so, again, that's just one criterion. There's a whole panel. The next couple of slides show another criterion for autism, and I'm certainly interested in getting feedback from our experts here because I am not a neurobiologist. But obviously mice can't talk, but the way that they communicate and socialize is through their sense of smell. And so what one can do is actually take a Q-tip, as you can see here, and coat it or embed it with various scents to see how the mice interact with that Q-tip as sort of a proxy for social interaction. And so first of all, they want to make sure, it's shown here on the left, that the mice have a normal sense of smell. So if they give them something that they like to eat, they do fine with that. But then, if you give a mouse a Q-tip that's embedded in the scent of another mouse, normally, as shown in these two parts of the figure, normally when you have a Q-tip with the scent of another mouse, that mouse is going to be interested in checking out who that is. And what they showed is that clearly in mice that are missing one or definitely two copies of the gene, they show almost no interest. So again, it's just one sort of surrogate for social behavior, but it's highly suggestive that these mice are not interested in interacting, or at least responding to sort of a cue for social interaction. Okay, so just to summarize, Jacobsen syndrome patients have variable degrees of cognitive impairment. We think at least two of these genes, BSX and neurogranin, contribute to cognitive disability. Deletion of that BSX gene in combination with one or more other genes appears to cause the most severe cognitive impairment, and we think that RIX is a strong candidate, at least as one gene, for contributing to the autistic phenotype. I should also say that even patients with the smallest deletions can have some mild degree of cognitive impairment, and typically it might be someone who goes through the regular school curriculum, but maybe they kind of struggled along the way, had some learning disabilities, special needs, but you know, think how many children there are that do that where we don't know the specific genetic cause. And I think it's very clear now that there are multiple genes in 11q that are contributing to these cognitive and behavioral problems, and also there must be other genes probably outside of 11q, not to mention other factors like environment, like the home situation, nurturing, early intervention, all those kinds of factors obviously that contribute. So in my history talk the other day, I showed you this slide, which I think is going to go down as one of the most significant Nobel Prize awards in the last bit of time. So this was in a recognition or an acknowledgement of the work to take differentiated cells and to be able to reverse their differentiation process to make them what we call pluripotent cells or stem cells. And again, this is not using embryos, this is taking your child's skin. And then to have the ability to reprogram those cells potentially into any kind of differentiated cell line that you're interested in studying. So obviously, what are we interested in studying? Heart and brain, for starters. So, and neural crests. So let me give you an example. This is from a collaborator of mine, an adult cardiologist, who has actually been able to reprogram skin cells to functioning cardiac myocytes. So that's the functioning cell for how heart beats. And just take a look at this. I think this is really pretty cool. These are cardiac myocytes that were reprogrammed, derived from skin cells. And this is just in a culture dish, but you can see they're not only beating, but they're beating in a very coordinated manner that really kind of simulates the contraction of the ventricle. So I think, for example, I don't know if Jeremy is here this morning, but we got skin cells from Faith Pyle, who has hypoplastic left heart syndrome, and one of the fundamental questions I'm very interested in asking is, do cardiac myocytes from a hypoplastic left heart have any fundamentally different pathologic properties that might help us to come up with better treatments. Similarly, we can reprogram these induced pluripotent stem cells to become brain cells. And what you see here, each one of these green signals is actually how neurons talk to each other. And so when I play this slide, don't blink, you'll see, if you look really carefully, little flickerings of these green signals, and this is actually a way that we can quantify how neurons interact, using calcium as a way to image. And so my collaborator here in San Diego, who is a neurobiologist at the Sanford Burnham Center, is working with me so that we can study your children's brain cells to see if there might be any differences. And as I mentioned, in order to do that, you have to be able to very comprehensively show that you can generate these induced pluripotent cells, and you have to really be compulsive to make sure that there aren't other alterations along the way that could fool you when it comes time to looking at the phenotype. And so we've done that on our first two patients. We've done that on Zion and Faith, and just to give you an idea of how expensive it is per patient, just to get to the induced pluripotent stem cell, it costs about $10,000 per patient. So we've got now over 30 patients, so do the math, which we would love to be able to do, but what I'm actually hoping to do is by focusing on a couple of patients that we think are going to be the most informative with a lot of different phenotypes that we're interested in studying, we can generate some important preliminary data that will allow us to come back and submit a grant proposal to study the much broader number of patients that you guys have helped us to want to study. And then you are going to hear a very, very interesting talk this afternoon by Dr. Shigani about the potential role of serotonin agonists for autism and how that might relate to your children. So I am out of time, and I'm done with my slides. There are a lot of people, some of them here in this room, that are incredibly helpful and supportive to make any of this work possible, and of course none of it is possible without you guys. So thanks again.
As you know, Vanessa has a small interstitial deletion.
Yeah.
And based on the location of that deletion, the end point is exactly that PXX gene.
Right.
Once the gene is knocked out, is she missing the whole PXX gene? Or can you just
that one copy is probably not functional. Yeah. She's basically missing one functional copy of the BSX gene. Yeah.
So it knocks out the whole BSX gene?
Right, so she has one normal copy. Yeah. But, as you know, Vanessa is relatively only mildly affected, and, that's why it's so important for our team here to do those assessments on her to kind of reproduce what you've already conveyed to me, and, that is a huge part of why when I specifically referred to locus 1, you might as well call it, Vanessa's locus, because that's what it's based on. Yeah, so Vanessa's famous.
Well, thank you for sending us stuff throughout the year as well. We read it, and sometimes we understand, like, when you say, hello, nice to hear, let's see you again, and then we'll watch for a while. I know that you did, you took these, and I'll say it wrong, pluripotent cells, and I think that, I recall, I think I wrote down that only 2 of 19 were successful.
Oh, no. So let me give you the exact numbers. So 2 years ago, we did skin biopsies from 22 Jacobsen syndrome patients at the conference. 21 out of the 22 were successful. The only one that was not successful was one that was a very small child that it was just too small of a specimen. We just didn't want to take a big chunk. So that one was just too small to grow up. So 21 out of 22 grew up, and of those 21, that's relatively easy and not too expensive to grow up the skin cell fibroblast cultures, which you can then freeze and store for as long as you want. Now, from those 21, I've gone back to 2 of those patients to make the $10,000 per patient investment. Yeah. So thanks for asking me to clarify. I wouldn't have asked you to do something if it were only 2 out of 21 that were successful. I would have been too embarrassed to ask you.
I think most of us will do, what do you need?
Well, I deeply appreciate that, but I don't take that for granted.
I'm pretty confident I speak for most of the room to say, you know.
Well, kudos to you.
I guess one other piece of information that would be helpful, at least for those of us who have older children, we forget what we've done. I don't know if you have a scorecard for all of us, but occasionally that would be helpful to say, you know, we could use this again, or you don't need that again.
Well, no, I mean, I do keep track. So, for example, you know, the skin biopsy is a one-time test. The deletion mapping should be a one-time test, although if it was done a long time ago, it's not going to have the resolution that the current platforms have now. So, but you know, at any time, I try to be as transparent as possible. And thank God for computers, and I could never remember all this stuff. But, you know, these days, I mean, we do keep good track. And Sarah and Natasha are also great. I mean, we go back to the records starting from their 2006 assessments, and you know, maybe it seems a little more chaotic than it is, but I can tell you, you know, from the scientific research standpoint, our living depends on being organized. Now, my wife would never believe that, but
well, I think we're not organized.
so I'm happy to share with you anytime. Yeah, just keep bugging me.
2 live stream questions. My son Dylan has JS and trisomy 9p. How do we know which of those is causing autism and ADHD?
Right, so that's a great question. Just so for those of you that didn't hear it, for those 5 or 10% of your children that inherited both a deletion as well as a duplication, the question is, how do you know which phenotype comes from the deletion of chromosome 11 and which phenotype comes from the duplication of some other chromosome? In this case, I think you said 9p. And the answer is you don't. And so that's why when I think Sarah presented her talk, one of our inclusion criteria for the very rigorous scientific studies is that it has to be only a deletion in 11q, because otherwise there's no way we can interpret if we do find a phenotype like autism, is it because of the 11q deletion or a 9p duplication? So that's why we have to be so anal about our inclusion criteria.
And second question, daughter's breakpoint is 24.1. She wanted to know if either the PSX or the milligram dystrophy.
Right, so again, 24.1, 23.3, those are not useful ways to assess the deletion breakpoint. What we have to have is the array data, and then I can look exactly at the readout to see which genes are affected. So that's the only way I can answer that question. So if that family wants to send me that information, if they can get it done, then I'd be happy to answer it. Any other questions? I think we can take like a 5-minute break and let Dr. Stein get set up. Great.
the picnic after this, just so in case people are...
You want to make an announcement now, just so people know what you got?
All right.
Yeah.